Welcome to this overview of Circoviridae and Anelloviridae, two families of small, non-enveloped, circular single-stranded DNA viruses. Circoviridae includes viruses like porcine circoviruses, beak and feather disease virus, and canine circovirus. They are non-enveloped, icosahedral, and replicate in actively dividing cells. Anelloviridae, including torque-tinoviruses in humans and pigs, are related but form a separate viral family, capable of persistent infections. Both families are widely distributed and diverse, making them important in veterinary and human health. Today, let's take a quick look at two important families of small DNA viruses, Circoviridae and Anelloviridae. First, the Circoviridae family. This group includes two main genera, Circovirus and Cyclovirus. Circoviruses are unique because they have an ambisense genome, meaning their genes are arranged in opposite directions on the DNA strand. Cycloviruses, on the other hand, are an emerging group, detected in humans, chimpanzees, and various other hosts. Common examples of circoviruses include beak and feather disease virus, a major pathogen of parrots. Porcine circoviruses, PCV1, PCV2, and the newer PCV3 and PCV4. Canine circovirus, and a wide range of avian circoviruses. Now, let's move to the Anelloviridae family. Within this family, the major genera include gyrovirus and torque-tinovirus. Gyroviruses are easy to recognize because all of their genes are oriented in the same direction, following a negative sense pattern. An important example is the chicken anemia virus, or CAV, a well-known pathogen causing immunosuppression in poultry. And finally, torque-tinoviruses are widely distributed and found in many species, including humans. Let's now look at the key virion properties of Circoviridae and Anelloviridae. These viruses are very small, measuring only 20 to 25 nanometers. They are non-enveloped, spherical, and have a simple T=1 icosahedral symmetry. Their genome is also extremely compact, only 1.7 to 2.3 kilobases of circular single-stranded DNA. In circoviruses, the genome is ambisense, containing two major open reading frames: the representative protein, which functions as the replication enzyme, and the cap protein, which forms the capsid and is the main immunogenic component. In gyroviruses, the genome is negative sense and polycistronic, with three ORFs: VP1, the capsid protein; VP2, a non-structural protein; and VP3, also known as uroapoptin. VP3 plays a key role in disease by inducing apoptosis in T cells, which is central to the pathogenesis of chicken anemia virus. One of the most important features of these viruses is their high environmental stability. They are resistant to heat, surviving 60 degrees Celsius for 30 minutes, and they show strong resistance to many disinfectants. These properties allow circoviruses and gyroviruses to persist in the environment and contribute to their ability to spread. That concludes the virion property overview. Next, let's focus on the morphology of circoviruses and gyroviruses, and how they appear in diagnostic samples. Within the genus Circovirus, the virions have a smooth, featureless surface. Their capsid is simple and uniform, consistent with their very small size. In contrast, viruses in the genus Gyrovirus, such as chicken anemia virus, have a distinctive appearance. Their capsid displays 12 trumpet-like projections at the vertices of the icosahedral structure. These projections are a key feature that helps differentiate them from circoviruses. Now, let's look at how these viruses appear in samples. Inside infected cells, both circoviruses and gyroviruses may be seen as free, individual particles. However, in cell-free specimens, they can sometimes arrange themselves in a pattern known as strings of pearls, a linear chain-like configuration of virions. This morphology can be a useful visual clue during electron microscopy or advanced imaging. And that completes the overview of their structural appearance. Virus replication, attachment and entry. Now let's look at how circoviruses attach to host cells and enter them. First, the receptors. Some circoviruses are able to hemagglutinate red blood cells, which suggests that they bind to sialic acid on the cell surface. For porcine circovirus 2, or PCV2, attachment is more complex. PCV2 binds to heparin, heparin sulfate, and chondroitin sulfate B, all of which are glycosaminoglycans present on many different cell types. This broad receptor usage helps explain PCV2's wide tissue tropism and its ability to infect multiple organ systems. Next, the target cells. Circoviruses show a preference for cells of the monocyte macrophage lineage, as well as dendritic cells and various epithelial cells. These immune cells play a major role in virus dissemination and pathogenesis. Now, for entry mechanisms. Circoviruses typically enter cells through endocytosis. For many strains, this appears to be clathrin-mediated, though alternative pathways may also be used. Entry into epithelial cells is particularly interesting. It depends on actin remodeling and small GTP-ases, but is independent of dynamin and independent of cholesterol, which is unusual compared with many other DNA viruses. These early steps, attachment, receptor binding, and entry, set the stage for successful viral replication and spread within the host. After entry into the host cell, the virus must uncode and release its genome. First, the uncoding step. Once internalized, circoviruses are trafficked through the endosomal pathway. Inside these endosomes, serine protease activity becomes essential. These enzymes help cleave the viral capsid, allowing the single-stranded DNA genome to be released into the cytoplasm and ultimately transported into the nucleus. Now let's move to genome replication. Replication takes place entirely in the nucleus and depends heavily on the cell being in S phase. Because circoviruses are so small, they must rely on the host's DNA replication machinery. The mechanism used is rolling circle replication, a process that begins at a stem loop structure located at the origin of replication. Two viral proteins play central roles, representative and rep. Both contain a set of highly conserved motifs that define rolling circle viruses. These include the RCRI, RCR2, and RCR3 motifs, essential for DNA cleavage and ligation, and a DNTP binding motif, which helps capture and utilize nucleotides during DNA synthesis. Together, these steps enable efficient amplification of the viral genome, setting the stage for capsid assembly and production of new virions. Now let's look at the key viral proteins produced by porcine circovirus 2 and chicken anemia virus. Starting with PCV2, PCV2 encodes only two major proteins: Rep, the replicase enzyme responsible for initiating and controlling viral DNA replication, and Cap, the capsid protein, which forms the virus particle and is the primary immunogenic protein recognized by the immune system. Next, let's review the proteins of chicken anemia virus, or CAV. CAV expresses three important proteins: VP1, VP2, and VP3. VP1 is the capsid protein, and it contains a unique 3-amino acid replication motif that is essential for proper viral replication. VP2 functions as a phosphatase, and it supports the replication process by assisting the correct folding and activity of VP1. And finally, VP3, also known as apoptin, is one of the most critical viral proteins. VP3 is essential for viral replication and is responsible for inducing apoptosis in lymphocytes, particularly T cells, a hallmark of chicken anemia virus pathogenesis. Together, these proteins coordinate replication, immune evasion, and disease progression. Let's now look at the pathogenesis of circoviruses and chicken anemia virus. Both virus families share an important requirement: they need actively dividing cells in order to replicate. Because of this, infection is more severe in young animals and during periods of lymphocyte proliferation, such as after immune stimulation or vaccination. This dependency on dividing cells also contributes to the development of persistent infections, although the exact mechanisms behind persistence are not yet fully understood. Now let's look more specifically at chicken anemia virus, or CAV. CAV shows remarkable persistence in breeding hens. Replication in the oviduct is enhanced by estrogen, which promotes vertical transmission of the virus from hen to chick. CAV persistence is also driven by the action of apoptin, the VP3 protein. By inducing apoptosis of lymphocytes, especially T cells, apoptin creates profound immune suppression. This weakened immune environment allows the virus to maintain long-term infection and enables secondary pathogens to take advantage of the host. Together, these processes explain the chronic nature and immunosuppressive consequences of infection with circoviruses and CAV.